This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode number 133, recorded May 13th, 2011. Hi, everybody. I'm Vincent Racaniello, and it's time for TWiV, the podcast all about viruses. I have a great crew with me today, starting up in Western Massachusetts, Alan Dove.
Good to be here.
You know, it's Friday the 13th.
Yes, it is, but I haven't had any particularly bad luck today.
Yeah, we were just recording.
We're working on it.
And the recorder just cut out, right, Dickson?
It did.
Oh, and speaking of Dickson, right across the desk, not in Fort Lee today.
Nope.
Dickson Despommier.
Hello, Vince.
Good to
Hello,
see
Alan.
you.
Hello.
Who else do we have here? Let's move down the coast
to
north central Florida and find Rich Condit.
Hi, everybody.
Hello, Rich.
How you doing?
Hey, Rich.
How have you been?
I've been good.
Yes?
It's really a good thing we record TWiV on a Friday.
Why is that?
Because I'm always in a good mood at 2 in the afternoon on a Friday.
Yeah, that's because of TWiV, isn't it?
Well, it does help. If you did this at 8:30 in the morning on Monday,
that would be bad.
Totally different podcast.
No.
I agree.
I don't envy anybody who has to teach on that hour.
Yeah, well, Friday's a good day to do this because
Yep,
Friday is a happy day. My kids are all happy when it's Friday.
Sure.
And Monday's a rough day.
It is.
Yeah, I agree.
You need a weather report. 91 degrees.
Right.
Ooh. No clouds, right?
A few.
Yeah, here it's all gray.
Yeah, it's summertime. We're
And,
doing summer now.
I don't know, what is it, 60-something, Dickson?
Yeah, not too bad at all.
Not too bad, but it's okay. And now for our final co-host, we move out to the middle of the country, Michigan, where we will find in Ann Arbor, Kathy Collins. Welcome.
Hi, thanks.
Hi, Kathy.
I thought we'd never get to you, right? we just banter and banter and banter. People say they like to hear scientists just chatting because they never get to do that. So that's what we do. And the only thing we can talk about is the weather.
Until we get to talking about viruses.
Yeah,
Exactly.
We've got
Mark-
sunny skies, almost 80.
Wow.
Oh, excellent.
Nice.
Very nice.
Nice.
Mark Twain said everybody's always talking about the weather, but nobody ever does anything.
That's
There
right.
you go. That's right. That's right.
Makes a big
Actually, though, we are kind of doing something about it, aren't we?
Oh, yeah, true.
That's right.
So, Kathy, you are in the Department of Microbiology and Immunology and the Division of Infectious Diseases, University of Michigan.
Yep.
An MD, PhD. So we're going to talk about AIDS and reservoirs of HIV today with you. But before we do that, tell us how you got to be where you are today. Where did you start?
Well, I was born in Massachusetts, if you want to go back
Wow,
that far.
Western Massachusetts?
Sure.
Whereabouts?
I grew up on the South Shore of Massachusetts in Norwell.
Wow.
Oh, okay. My sister lives down in Swansea.
Okay. Yeah. So I grew up there, and then I went to Wellesley College, which is also in Massachusetts.
Yep.
And that's when I had my first experience in a research lab. And I was studying interleukin-1 with my professor named Andrew Webb, and I got really excited about science and decided that I was going to pursue that as a career. I had up to that point just thought about being a doctor, and I had already been accepted to medical school at that point. So
Oh, wow.
I deferred, stayed on in the lab for a year, and then applied for the combined degree down at Johns Hopkins.
Wow, that's heavy duty. You had already been accepted.
Yeah.
Right.
Wow.
My
Cool.
parents were a little worried.
That's right. You run off and join the circus, maybe.
So they allowed you to defer at Hopkins with the
Yeah.
idea that they would take you in a year? That would be okay with them?
Yeah, they were fine with that. And then during that year, I was able to get into the MSTP program down there. So I had some funding to get the PhD degree along with the MD degree, which
Right.
Terrific.
helps a lot.
So let's tell people what MSTP is.
So MSTP is an NIH funded program, and its goal is to allow MDs who might otherwise be tempted to go into private practice to pursue an academic career, and they help them out with that by providing some funding so that they don't feel so much under financial pressure to make money once they graduate, and they can be freer to pursue academic goals.
So MSTP stands for Medical Scientist Training Program. Is that right?
Yes. Yeah.
Okay.
Yeah, we have one here. You probably do too, Rich? Right?
Not an NIH-funded one.
No.
We have an MD PhD program, that is working on trying to get enough cred to get an MSTP grant.
So, Kathy, after you finished your MD PhD, did you do a postdoc or a fellowship?
I started out with an internship
Right.
in the Boston area at Brigham and Women's Hospital, and then I did a residency in internal medicine and a fellowship in infectious disease. Then I had to go and do a postdoc where I went over to MIT for that in David Baltimore's lab, and that's when I started doing HIV work.
I didn't know you were a Baltimorean.
I am, as I know you are.
Yeah, the more recent ones I don't know.
Yes,
I don't know if you overlap with Shankar Ghosh at all.
No, I did not. I was there in 1996.
Yeah, he's our chairman here now. You probably know the name, right?
Yeah, oh yeah, definitely.
Did you overlap with Rebao Ren?
No,
okay.
No, I didn't.
So I was there from, you guys have to pardon us.
It's okay.
We'll just talk among ourselves.
I was there from 79 to 82. We were still at the cancer center at MIT.
Yes.
But you were at the Whitehead, I presume, right?
Actually, we were in the biology building, the new biology building. This was after David came back from Rockefeller.
Mm-hmm.
He was at in the biology building for a few years before he went off to Caltech and became president there.
Okay.
Yeah, I knew him when he was a student at Rockefeller.
Is that when you were there at the same time?
Yeah.
Really?
From 64 to 67. 67 to 70, sorry.
You were there that long ago? You must be an old guy.
I'm an old guy.
Wow. So, Kathy, where did your interest in HIV come from?
Well, when I was in Baltimore doing my MD PhD, I was in Tom Kelly's lab.
He's here in New York City,
So wait a minute. You went from Baltimore to Baltimore? Let me get this straight here.
Yes, you did. Tom Kelly is here at Sloan Kettering, I believe.
Yes.
Yep, he's the head of the whole joint.
Yeah.
That's right. So I knew him when he was just chair of the department at Johns Hopkins. And
back before nobody had heard of him. Yeah.
Yeah, that's right. And we were studying SV40 DNA replication. And during that time, it was around the late 1980s, HIV was hitting Baltimore really hard. And
that's the city of Baltimore.
The city of Baltimore, yes. And I was, going back into medical school, and we'd see 1 in 10 people coming through the emergency room being HIV positive, and it was just, the virus of the age, and
sure.
So it was a compelling disease to study, and so timing wise, it made sense for me to pursue that.
Did you know Al Saw?
No, I did not.
Okay, he was at Johns Hopkins.
Okay.
So, did you work on HIV in the Baltimore lab?
I did. I don't know if you've run into Ben Chen.
Sure. Yep.
So Ben was a graduate student in the lab when I joined, and he was getting these marker HIVs with a placental alkaline phosphatase gene expressed, so that he could monitor cells by flow cytometry and find the infected subset of T cells. And that was a really useful tool for what I wanted to do.
This must be before GFP or something like that.
This was before GFP, yeah.
That's
right
the kind
around
of thing.
the time.
That's the kind of thing you do at GFP nowadays.
That's true.
In fact, that's what she's done in these nice papers we're going to talk about. Yeah,
that's right. And
because you can't do flow very well with the phosphatase, right?
Well, we would use antibodies actually
against the phosphatase. Yeah,
the alkaline phosphatase. It's a GPI-linked protein, and that's how we found the infected cells.
Did you go to Michigan right after your postdoc then?
Yes. Yep.
So you have a joint appointment in ID, so I presume you see patients as well?
I do. For about a month a year, I see patients at the VA hospital here in Ann Arbor.
So that's a typical duty of a clinical appointment who's doing research, right?
Yes, that's pretty standard, actually.
Yeah.
So a month a year, you leave the lab and do that full time, or you how do you divide that? Because that always seems to come up with people who do that sort of thing.
Yeah, it's a little tricky, but I'm usually able to be in my office here near the lab in the mornings and touch base with people who are doing experiments, and then in the afternoon I go around on the patients with the clinical team over there, and it works out pretty well. It's busy, but it for a month a year I can do it and feel like I'm doing a good job still.
Have you noticed a gradual decline in the number of patients with HIV in your hospital?
It's been, there's definitely a decline in admitted patients.
Mhm.
The clinic has remained fairly stable as long as I've been here. We have about 600, and they're healthy and they're outpatients.
Got it.
They don't get sick so much anymore.
Exactly. And they're all veterans of the armed forces, I take it.
Yes. So
VA hospital.
Yeah, but sometimes family members can get into that as well. Sorry to interrupt.
No, it's okay. We do that all the time, as you know. So I think we have talked a lot about HIV and AIDS on TWiV, right, guys? And we probably don't need to give any background. But what I wanted to talk about with you, so we have a couple of your articles studying the reservoir of HIV in people. So tell us a little bit give us some background on why we need to know about a cellular reservoir.
So as we just mentioned, people who have HIV disease are really healthy now because of highly active antiretroviral therapy, which is often called HAART. And HAART is a very effective combination of usually 3 drugs. And in most infected people, a regimen can be put together that leads to declines in levels of the virus in the blood, so-called viral loads. And the viral loads are so low that they can't even be detected by the clinical tests that are standardly used to quantify how much virus people have in their blood. But despite years and years and years of treatment, when people stop taking their drugs, the virus comes back.
Right.
And this was initially a surprise. I think people were hopeful at first that if people were treated for a few years, the virus would be gone, because at the time people thought that most virally infected cells lived for short amounts of time, that the virus was pretty toxic to the cells, and even longer lived cells had a shorter half-life when they were infected and the virus was replicating. And so it was a bit surprising when, after years of therapy, people could not come off their drugs. And there were 2 ideas, and there still are 2 ideas, or at least 2 ideas. One is that the virus is still replicating, but it's just replicating at such low levels that we don't detect it. And so in that model, the cells are continually being infected and dying off and making new virus, and the virus spreads to other cells. And then the other model is that because HIV integrates into the cellular DNA, if the viral gene expression is not turned on, the cells will live their normal lifespan. And at some rate, the virus could decide to turn back on and make new viral particles.
What causes that to happen?
So that, which is called viral latency, can be caused by a number of things. One is that the cell that has the integrated viral genome isn't making the right proteins that the virus needs to turn on its promoter. So NF-kappa B is one of those proteins, and not all cells express enough of it to turn the virus on in the state that they're in. But the cell could become activated, or it can be induced to differentiate into a different type of cell that expresses higher levels of the protein, and then viral gene expression can be turned on.
Right.
So the two models are either it's hiding everywhere, but at such low levels that you can't find it, or it's hiding in a specific compartment, but it's turned off until some environmental cue turns it on.
Yes. Yep, that's right.
If you have a mixture of, I'm trying to figure out an in vitro experiment to do this. I'm wondering if in culture you can actually cure a culture of HIV. Because Model 2 seems just intuitively correct to me that because the virus exists as a provirus, that there's always going to be cells around that are making a little enough virus so the drugs won't touch them, and you have a copy of the genome somewhere, so that if you withdraw the drugs, that guy eventually is going to reactivate and cause a problem.
I think people are leaning towards that model. There have been a number of papers published in which people have done so-called intensification experiments, where they've added another drug, or they've added drugs that get better penetration into certain areas that are hard to treat, like the brain.
So
they've looked to see whether the reservoirs change, and they haven't.
So the cells harboring the provirus are long-lived cells?
That is the thought, that they would need to be long-lived cells, yes.
Before HART, we didn't have any sense of there being a reservoir?
Well, it's interesting. The idea of latency in HIV disease has evolved over time, and it used to be that when people didn't have symptoms, the thought was that the virus was latent.
I see.
And then we learned that in the lymph nodes, the virus is actually continually replicating, and there's a lot of active infection. And then we started measuring virus levels, and we could see that they were variable in people, but usually detectable. And so then we realized that period of clinical latency was not actually virological latency, and so latency went out of favor for a while. And then with the advent of HART, people got more interested in whether or not there was actual virological latency at a really, really low level that probably doesn't matter with natural disease, but in this situation in which we're getting rid of everything else, the low amount of latency that is achieved by the virus now takes on a lot of importance.
So you're making a distinction between, I always have this problem in teaching this stuff. You're making a distinction between clinical latency and virological latency. Is that right?
Yeah.
Something like that. Because latency is a kind of a squishy word, it seems to me. Yeah. I always have trouble with. I sometimes make the distinction by calling it molecular latency or something like that, where in fact the genome is there, but it's really not doing anything.
Right.
That's true molecular latency, or what you're, I guess, calling virological latency.
Yeah. Yep,
as opposed to maybe you're making a little bit of virus or something, but it doesn't show up clinically. You can't measure it. There are no symptoms.
Yeah, that's
none of which has anything to do with Freudian latency.
No. Alan. Alan's
just thought I'd point that out.
He's a bit of an outlier, but he's a friendly, wonderful outlier.
You got that from your wife.
So I need to ask, are there no therapeutic strategies invented in the past for other viruses that become latent that you could actually identify them in the genome and eliminate those cells by another technique rather than using the triple therapies that are used?
Yeah, people are racking their brains trying to come up with some strategies like that.
Yeah. Because I think about herpes a lot and other diseases that are latent as well. And
can't get rid of those either.
Yeah, wouldn't it be wonderful if you had an agent that identified the cells with the integrated genomes and just killed them?
Well, if you remember, we've talked, and Kathy, you can chime in. Someone asked us about a paper using tree recombinase that had been evolved to recognize HIV LTRs. I don't know if you've seen that or not, but in cell culture, at least, they can excise proviral DNA using this enzyme.
I'd forgotten about that. Yes, that's cool.
A strategy like that would be great if we could effectively get it into enough cells.
Yeah, that's the problem. You have to get it into the right cells, and also you can't have off-target effects.
But Dickson's comment is appropriate. This is an exam question that I use for herpes, is asking people whether or not the antivirals for herpes could cure a latent infection, right?
Yep.
And they can't because, they don't act on the genome.
Sure.
They
That's
act on the transcription.
In fact, we had the same question on my virology exam this week.
And
All right,
just to
great.
add more fuel to the fire, there's a eukaryote called toxoplasmosis, which exists in a latent form also, which doesn't undergo division. So the drugs that interfere with the division of the organism won't work, so they can survive forever in your brain like that.
It's very hard to target an organism that's not doing anything.
See that?
Right.
Well, that's what I'm driving at. So how much investment of time, Kathy, do you think the NIH has put into funding research to kill off cells that have inactivated viruses?
It's now a focus.
Good.
It's now a focus. I think they've recently put out a couple of program announcements and some program project initiatives to try to get people focused on this question. And most of the program project initiatives, interestingly, ask that you find a drug company to work with.
But of course, like Park Davis, for instance. No, we won't mention names.
Well, I think we've seen the news about the current NIH budget, though.
Yes.
Yes, that's true.
And this is a reasonable target for trying to collaborate with industry.
I mean, you could see, you could get a little snippet of genome from the virus connected to a protein that has access to the nucleus, and then there's a toxin associated with that too. When they all three come together, the cells go.
Write a grant, Dickson.
No, I'm not. I'm
you're
retired.
You're retired. Okay, so Kathy, how did you start? We're going to talk about your identifying these long-lived cells. So how did you get started on that?
Well, we had been studying one of the HIV proteins called NEF, and NEF functions by helping HIV evade the immune system by down-modulating a protein called MHC class 1.
What does NEF stand for?
Negative effector factor, which is
See, ask the dumb question, you get a dumb answer. Sorry.
Yeah, it's unfortunately a misnomer. People had thought that expression of NEF was somewhat inhibitory to infection, and so it got this name.
Most proteins end up getting named before anybody has any clue what they really do.
Yeah, and some pretty bizarre names at that.
So we're stuck with NEF, but it's a really interesting protein, and it disrupts antigen presentation, which makes it harder for the immune system to find actively infected cells.
Is it a surface protein?
It's it's linked to the membrane by a fatty acid that's inside the cell, except it's linked to the membrane, and it binds onto the cytoplasmic portion of MHC class 1 molecules and disrupts their transport to the cell surface.
Cool.
Amazing.
Right, so the cell can't indicate that it's infected.
That's right. It has it's defective at that function. And so then the immune cells need to see MHC class 1 in order to find the cells, and they have a harder time finding them when NEF is expressed.
Wow.
So we'd like to develop inhibitors of NEF, actually.
Of course.
And, that might help the immune system find some low-level infected cells and help clear these cells from the body. But
which molecule of the MHC does it bind to, by the way?
It binds to the group of MHC molecules called HLA-A and HLA-B.
Got it.
It doesn't bind to HLA-C, which is another really interesting story that could take a whole show. I think.
Okay, that's a deal.
Yeah, there's a lot of interesting new information about the role of HLA-C, which is not affected by NEF. Mary Carrington would be a great person to have on your show.
So drugs that bind up NEF so it can't combine with its ligand would be a great drug.
It would help the immune system clear the infected cells. And none of the drugs we have now are active against the infected cells. They only target the virus particles.
Do any other viruses elicit NEF?
No, but a lot of these chronic viruses, like herpes and cytomegalovirus, they have proteins with similar functions that disrupt antigen presentation.
Because they all face the same problem.
They all face the same problem. That's exactly right.
Interesting. Fascinating stuff.
So we were interested in how NEF worked, and we were studying it in T cells, but we also wanted to understand its role in antigen-presenting cells, which is another target of HIV. And we noticed that when we put the virus into macrophage precursors, that we didn't get very much viral gene expression until we differentiated them. And we thought that maybe we were looking at a kind of latency. And so we kept going into more primitive progenitors and seeing the same thing.
Got it.
And eventually we went to bone marrow and cord blood that have the most primitive progenitors, and we saw something similar, that we could infect them with the virus, which we were surprised about, and also that when the cells were infected, some of them became actively infected, but a good portion of them became latently infected, and we could reactivate that latent infection by causing them to differentiate.
So these are cells from, as you said, either human bone marrow or human cord blood, right?
Right, yeah.
Which are easy to get, I presume?
You can purchase it from companies, actually.
Does Irving Weissman make it for you?
No.
You can buy it. Okay. So I wonder if you could explain briefly. They have a lot of different kinds of precursors, multipotent hematopoietic stem precursors. Then you have just hematopoietic stem cells, multipotent HSCs, MPPs, CMPs.
Yes.
Could you set it all straight?
Well, it's complicated. In the bone marrow, a minor fraction of the cells there are so-called progenitor cells, and it's really less than 1% of the bone marrow cells are progenitor cells. And within that 1%, a small fraction, maybe another 1%, are stem cells. Mm-hmm. And stem cells have a lot of really unique qualities, and they have the capacity to self-renew, and so they can make more copies of themselves. And they also have the capacity to generate daughter cells that can go on and differentiate into all of the different kinds of blood cells that we have. So T cells and B cells and macrophages and phagocytic cells, granulocytes, blood cells. So they're very powerful.
What are those called? Those daughter cells?
Those would be the MPPs would be the
multipotent,
slightly more differentiated, and then there's common myeloid progenitors and common lymphoid progenitors. And each step along the way, they're a little bit more differentiated, and they have a bit less of a capacity to self-renew and to differentiate into other types.
And none of them revert back to stem cells at that point?
Not naturally.
Can they be induced to do that?
I think this isn't my area, but my understanding is that we can take cells and make them go backwards these days.
Mm-hmm. Right.
So when you look for infectability by HIV, you look at all these different kinds of cells. You look at the precursors and then the different kinds of progenitors.
We do. It can be hard to tell them apart. Mm-hmm. We use a technique called flow cytometry, which allows us to see cell surface markers. And since we have markers inside the virus, we can find the virally infected cells and see what kind of cell surface markers they have. And that helps a lot. There's a marker called CD34, which is specific to progenitor cells, but it can't tell you whether the progenitor cells are multipotent or stem cells or committed. And so you can use more markers, and they help a bit more. But really have to go to functional studies to really tell what their developmental capacity is.
So when you do your studies, do you typically take, say, cord blood and then purify all the CD34 positive cells from it?
Yes.
And then
we start with that.
And then you infect them. So and you use viruses that have GFP in them, right?
Yes, that's right.
So that you can look at both CD34 and the virus at the same time.
Yes.
And I think that's in one of your papers you mentioned that this really helped advance the field because previously there was always some kind of uncertainty whether the CD4 positive cells were the ones that were infected.
That's right. And, I should say that, we sort of reinvestigated an old question. You can look back in the literature into the early 1990s when a number of scientists also published reports that HIV infects these precursor cells. And there was a lot of controversy, and other people said, no, they don't. Other people said, yes, they can. And it remained sort of ambiguous, and I think a lot of people ended up deciding that it wasn't very important if it was happening. And we reopened it now in a new era when low-level infection of long-lived cells is taking on a new importance. It's probably true that these progenitor cells don't contribute in a major way to high viral loads in untreated people. Mm-hmm. But we think there's a reasonable chance that they do contribute in treated people to residual virus that could reinitiate an infection when drugs are stopped. Mm-hmm. And we're investigating, that question.
Right. At the very least, you found some cells that do. You have not necessarily found the only reservoir, but you found some cells that look an awful lot like one place that HIV is hiding out in people who've been treated, right?
Exactly. And I should mention that a much better characterized reservoir is the resting memory T cell. And a lot of people have done some great work characterizing that reservoir. And the resting memory T cell reservoir is very interesting, too, because these cells are long-lived and probably have some capacity to self-renew. And that's why people have vaccine responses that can last their whole life because of the resting memory T cells. And so virus that integrates into this population of cells can also live for a long time and become reactivated.
It's amazing how viruses have become the world's greatest cell biologists, isn't it?
They are. Are the memory T cells as long-lived as the hematopoietic progenitor cells?
Yeah, that's an interesting question. When people have done half-life analyses, I've seen numbers put out there that they live for, that their half-life is 44 months, for example. I don't know how accurate those kinds of estimates are. I do know that people can have vaccine responses that, can last well into old age. And so I would expect that at least some of them can live quite a long time.
But the progenitors, the hematopoietic progenitors, basically can live the life of the human, right?
They can live the life of the human. That's right.
So in your Nature paper from last year, you showed that if you took these HPCs from cord blood, they could be infected with HIV, and you had to use HIVs that would not kill them, right?
Yeah, that was important because we had to use functional assays to characterize them and show that they were actually progenitor cells. And if we put wild-type virus in it killed them before they could do anything. And so we used gutted HIVs, HIVs that had those toxic proteins removed.
Right. So in a person, if this were the long-lived reservoir, what would prevent virus in them from killing those cells?
It would be latent. And so the proteins, the toxic proteins wouldn't be made. The virus DNA would just be hiding in the genome, ready to be reactivated.
But you never see that in culture, right?
We do, actually. A proportion of the cells are latently infected. And
what fraction, roughly?
It varies. I would say maybe between a tenth and a third on a typical experiment.
So how do you define experimentally? How do you define latency in that case?
There's a couple of different ways we've done it. The actively infected cells die off because the virus is toxic. And so if we culture the cells for, say, a week, we don't see evidence of active infection anymore. Then we can take those cells and treat them with cytokines to induce them to differentiate down the myeloid lineage. And under those conditions, we've shown that we upregulate NF-kappa B, and the viral promoter turns on, and we start getting viral proteins being made.
Which interleukins do you use?
We use GM-CSF and TNF-alpha.
Right. So go back to your Wellesley days. And interleukin number 1. How many interleukins are there now?
I don't know. I've lost track.
Exactly. No, that's my point. We've all lost track. It's up in the 30s, I think, and it's quite amazing. Since I represent a field that no one pays attention to, the eukaryotic parasite field, that's just a joke. A lot of those interleukins were learned about.
Keep your day job.
I don't have a day job anymore. A lot of the interleukins were not known until we started to look at the immune mechanisms against things like Leishmania and Toxoplasma. But now they can feed back into these other viral infections also as important players with regards to the pathogenesis of a multiple number of infections. I think it's fascinating, absolutely fascinating.
So I want to understand this culture thing better. You said you can have cultures of infected cells. The cells are killed, so basically you'll have a virus-free culture eventually, right?
That's right.
If you passage these. So are those cells that came from a patient or that you infected yourself, or can you do it either way?
We can do it either way. It's hard to do it with patient samples because being able to see this requires a high rate of infection. And often the rate of latency from donor bone marrow is so low that you can't see it by this assay, and you have to use more sensitive techniques like polymerase chain reaction to amplify out DNA genomes.
So at any rate, so you passage these things for a while, and you can get a culture that's genuinely virus-free.
Yes.
All right, but then you can induce those, and they'll throw off virus. So this is going to come down to understanding the molecular details mechanistically of that latency. Mm-hmm. Obviously, NF-kappa B is implied.
Yes. Yep.
Yeah.
So is it a reasonable strategy so if we assume that the HPCs are a major latent reservoir, can you then activate them in people and hope that they lyse and that's the end of the virus?
So that's an interesting strategy that a lot of people are trying to make work. They call it sort of the flushing out strategy, where they come up with chemicals or cytokines that will turn on the viral promoter, make the HIV proteins, which in turn kill the cell, and all the while preventing new virus spread by using the highly active antiretroviral therapy, hoping that the virus will kill the reservoirs off.
So if you had an identical set of twins, and one of them had HIV and the other one did not, and you cured the HIV patient with triple therapy, and then irradiated their bone marrow to kill off all the cells in the bone marrow, and transplanted the bone marrow from the other twin into that person, you could cure them entirely of their infection, theoretically.
I'm not sure. It's a possibility. There was, I don't know if you guys have talked about this German patient.
Yes, we have.
Yeah.
The German patient. there was an English patient. I know the book, but I'm not familiar with the German patient.
So there's one case report in the New England Journal of Medicine where a person who was HIV infected also required a bone marrow transplant because they had cancer and they needed it for that. And they went through a course of chemotherapy and irradiation to prepare them for a bone marrow transplant. And they were transplanted with bone marrow from somebody who had an inherent resistance to becoming infected. They were genetically defective in one of the HIV receptors, CCR5. And this individual seems to be cured. My understanding is that they've been several years off therapy with no signs of recurrence. But that's a very unusual situation.
Yeah. So cancer could cure HIV.
Well, then you can cure the cancer.
Right.
It's kind of that's why I asked you about Al Saw, because Al Saw was at the School of Public Health at Johns Hopkins.
I see.
And he uncovered a nest of people, a nidus of people that were totally resistant to HIV, about 5 of them. And he was in the popular press for quite a while because of that, suggesting that there's some limitation to the number of people that can actually become infected. So under natural selective pressures, Forget about the drugs. These would be the survivors.
Of course, a bone marrow transplant isn't a good way to
No, of course not.
cure AIDS, but the point is that if you can get these long-lived cells to multiply and be killed, that would do it.
Exactly.
Right.
So-
What you need is some better strategy to flush the virus out of these cells, which you have to be careful with because you do actually need hematopoietic precursor cells.
Yeah, so that's why I wanted to ask Kathy, what fraction of your precursors would be latently infected? Do we know?
So far, our estimates are probably less than 1 in 10,000. Is what our current estimates are. So, if you could just get rid of the latently infected ones, you would still have a lot left.
But you said the long-lived memory T cells are still a problem.
Yeah, you still have those,
Problematic.
Right.
Kathy, to what extent can you model this in an animal? I notice you've got some xenografted mice or something. To what extent can you?
Please
model
explain.
this?
Well, we did use mice, but we didn't use them as a model. We used them as an assay because I said it's hard to tell whether a cell is a stem cell.
Right.
Mm-hmm.
And the gold standard is to show that the cell can engraft in the bone marrow and generate all of the lineages.
I see.
Yes.
That requires both self-renewal and pluripotent differentiation into all these different cell types. And so we worked with Sean Morrison here to do that experiment. He's the director of the Stem Cell Institute here at Michigan, and he helped us with these experiments. And basically we injected the cells into the bone marrow of this immunocompromised mice, and the cells were able to engraft and generate T cells and B cells and macrophages. And that was evidence that at least some of the cells that we were targeting with the virus were stem cells. And
those were those HIV infected stem cells?
They were. We used a gutted HIV.
Okay, right, GFP, so you could monitor them.
Exactly.
Okay.
Exactly. And they were. There was an HIV envelope that allowed the virus to get into the cell.
So that's another proof that these are multipotent stem cells that are infected, right?
That's right.
And beyond that, you don't really have an animal model to study this, right? You're stuck with using patient material or studying some of the aspects of this in culture, correct?
That's what we do. We get patients to donate their bone marrow, and we isolate the progenitor cells and look for evidence of HIV and try to reactivate it from those samples. But there is an animal model that's reasonable, and it's using monkeys and macaques
with SIV.
With SIV, yeah.
So they get latent infections as well, and is it the same progenitor cells that are latently infected? Nobody's
done that. Nobody has at least published any evidence of that so far. But it's still early days. It'll take some time to look.
So in your other paper, you identified CXCR4 as the co-receptor for HIV to infect these progenitor cells that we've been talking about.
Yes.
So what's the significance of that? Because we know there are two co-receptors, right?
That's right.
So these are only infected via this one co-receptor.
Yeah, it's really interesting. It seems fairly conclusive. We can get some infection by the virus that uses the CCR5 co-receptor, but the progenitor cells are farther along the differentiation pathway. And to target the most primitive progenitor cells that are more stem cell like, we need to use a virus that utilizes the CXCR4 co-receptor. And our evidence is that it's because the cells that are more primitive don't express the CCR5 receptor to the extent that they express the CXCR4 receptor. And it has a lot of implications because, for one thing, early in infection, most of the virus that's made is CCR5 tropic. So one possibility is that the bone marrow reservoir isn't established until later in disease, when you start to see more CXCR4 tropic virus. And we're going to start some studies to look at that. But it's also possible that because HIV is what they call it's a quasi-species. It has a lot of variants because it can mutate. It can mutate fairly rapidly, and even though the CCR5 tropic virus predominates, there may be enough CXCR4 tropic virus early in disease to establish a compartment where you can find this virus in the bone marrow. So we're going to be looking at that to see whether that's true.
So does that mean that if you treated someone early enough with heart before these R4 using viruses arose, you might prevent infection of the progenitor cells?
Yeah, that's a possibility. They might still have the resting memory reservoirs, but they may not have the bone marrow reservoirs at earlier stages.
So this is where you need the appropriate animal model, where you could actually infect an animal and
do time courses.
Do time courses to see when you establish these reservoirs and whether or not, if you treat with drugs soon enough, you can actually affect a cure.
That's a lot of monkeys.
It's a lot of monkeys.
That is well.
What we're doing is we're trying to recruit some donors who were treated since acute infection. Some people have symptoms during the early stage.
Yeah, I was going to ask how difficult it is to identify people who are newly infected.
It's not common, but it happens. And if those people are treated right away, we can get their bone marrow and look at it and see whether they have reservoirs established early on. And that's an interesting question. The other implication of knowing that it's X4 tropic virus and infects these cells is that it's long been known that that even though R5 seems to be more important initially, that as time goes on, the virus tends to evolve towards an X4 tropic virus. And when that happens, it's associated with a worsened clinical prognosis. People tend to have more damage done to their immune system and to progress more rapidly. And so one idea is that when this X4 tropic virus evolves, it's now able to attack the bone marrow as well as the T cells in the blood, and that might make it more difficult for the bone marrow to replace the CD4 positive T cells that are being attacked in the blood. And the combined effects on the bone marrow and the blood might lead to reduced ability to fight off the virus.
So does this conversion happen in every infection from from R5 to R4?
It happens in at least 50% and probably more.
Okay, so the ones where it doesn't happen, so you have R5 utilizing viruses for the entire infection, do they have latent, long-lived latent reservoirs?
We haven't looked at enough people yet to really say conclusively. We have one example of a donor where we could only find R5 virus in their blood, but we were able to find X4 virus within the bone marrow cell.
I see.
Now, when it transmits to the next host, if the infection always follows this course, there must be some selective pressure early on in favor of the R5 and some kind of pressure that later on favors the R4. Any idea what that might be?
No, it's a really fascinating research question. A lot of people are interested in understanding what are the bottlenecks to the virus when it infects, and why R5 is selected at that stage, and it's really not well understood yet.
Do you think it's the bone marrow?
I think
Or maybe the initial immune response.
Yeah, I think it's hard to know why R5 seems to do so much better than X4 tropic viruses early on. I suspect there's some restriction to X4 until the immune system is damaged, but and then it's allowed to grow out. But it's not well understood.
So we've talked entirely about the hematopoiesis system. Is there any evidence that HIV can establish latency in other tissue types?
There's some evidence that the brain and the cerebral spinal fluid can be a source of virus, and there the story has more been one of low-level active infection, is my understanding, because the blood-brain barrier sometimes prevents drugs from achieving similar levels in the cerebral spinal fluid as it does in the blood. And the lower levels of drugs in the brain may allow the virus to replicate to a degree.
Okay. So I'd like to ask you a general question, if you don't mind, because I am old enough, as Vince pointed out so aptly, to have been a professional parasitologist during the advent of the AIDS epidemic. And I would like to know, from your perspective, what do you think the biggest changes have been since we originally detected this virus and studied it and then characterized it to the present time? So it's 1980, let's say 1982 to 2011. In your view, what are the biggest changes that the virus has undergone since the initial characterization?
What you mean, has the virus evolved and changed?
Exactly. Exactly. It can't be the same that as it was in 1982, though, right?
Well, the sequence of HIV does evolve within an individual. The virus that they have after 10 years is different from the one that infected them. It evolves in response to the immune system pressures and also to drugs that are initiated. The virus will evolve to try to become resistant to those drugs. And similarly, from person to person, the virus is different. And so the pressures that we put on the virus will determine what the virus becomes. And so there's more virus that's resistant to drugs that we've used for a long time.
Sure.
And so these days, when somebody is initiating treatment, it's fairly common to actually sequence portions of the virus to find out whether resistance gene mutations are present in a person before drugs are initiated.
Got it. But they are employing the exact same receptors for gaining entrance into cells in 1982 as they do now, or has that actually undergone modification also?
That's a good question. Back then we didn't really understand that there were two different receptors. And so, we weren't thinking about it that way. But I would imagine that particular phenotype would be a constant.
Right.
Yeah, the virus has obviously worked out a successful strategy.
They tend to do that. Nothing like selection, right?
It's true. Kathy, do you know Scott Hammer? He's our HIV expert here at Columbia.
Okay, I've heard of him. I've never had the opportunity to meet him, though. Okay.
Yeah, he does a lot of clinical trials.
Indeed.
Vaccines and used to do antivirals.
Kathy, were you equally disappointed and disillusioned over the latest failure of vaccine development from the NIH? Were you as devastated as Tony Fauci?
It's a really hard problem. The vaccine businesses, and it's such an important one because, in this country, we can afford to put people on drugs and keep them healthy, but it's harder to do that in developing countries that bear a bigger burden of the disease. than we do, and it's getting somewhat better. They are putting more and more people on drugs, but it's because it's a lifelong commitment to therapy. It's really financially difficult to sustain, and obviously a vaccine would be hopefully a one-shot thing, and people would be protected. So it's still a really important goal that we need to continue onward on.
That's interesting. I was just thinking that actually, if you could get everybody on drugs, since the
If you didn't Timothy Leary try
thanks,
that?
right? Since the drugs knock the replication of the virus down to the point where it's basically undetectable, that must interrupt transmission, right?
Yep, it does. Yep, there's much less transmission when people are affected.
So if humans are the only reservoir, you theoretically could eradicate the disease just with a drug treatment.
If you waited long enough, yeah.
Mm-hmm.
But you still have those latent viruses, Rich. If you stop taking your drugs, they'll come out.
Yeah, but you got to. It's going to take a generation. All the infected people have to die and not transmit it.
But they won't all keep taking drugs for their whole lives.
Yeah,
a good fraction, right?
I said theoretically.
Oh, I missed that.
Right. So I want to ask another question then. Does Magic Johnson support your research at Michigan since he was a star basketball player for your school?
I haven't heard from Magic.
I mean, he's one of the longest celebrity survivors for HIV, I think.
Yeah, he is.
But he's no different from non-celebrity survivors. Dickson.
No, but the fact that he went to the University of Michigan.
I understand that.
I thought that maybe...
And that he's a celebrity. Maybe he could,
And he's not poverty-stricken either.
I haven't heard from him lately.
Which countries...
So, Magic, if you're listening, give Kathy a call.
Right. Kathy, what are the countries that you're most worried about as reservoirs for the infection itself without going into too much details? Which are the countries that need it most?
Sub-Saharan Africa is probably,
all of it.
Yeah.
Except Senegal.
30 million infections.
Yeah.
It's huge.
And Russia?
Who knows?
Yeah.
Okay.
Hey, Kathy, I want to ask you, what do you think about XMRV as a human pathogen?
Interesting question. I'm not sure I want to comment on that.
How's your email box?
Yes, it'll fill up rapidly if you do.
Yeah.
Have you paid any attention to it?
I have. I think it's fascinating. It seems like for some populations maybe, but maybe not for others. I don't know. What do you guys think?
Uh-oh.
We're waiting for more data.
None of us think, so we don't have a problem with that.
There are a couple of papers that are coming out. So actually, one just came out last week, which we haven't yet talked about on TWiV, and there are two more, I think, in the pipeline and I think we'll do them all together at some point. And then, of course, there's Ian Lipkin's study, which is due out at the end of this year, which may really resolve everything, but
is that right?
I'm not sure. It's a very interesting story and how it's been handled by both sides. I mean, the virologists have not been great about it because whenever there's a result, they say, oh, so this shows that it's all nonsense. And then the other side gets mad. And, I think it's a good lesson looking backwards on how to handle this. But my personal view is that it's isn't really resolved one way or the other. We have some interesting information, but the one group that has supposedly the most information, this institute in Nevada, doesn't publish very often. So it's very hard to tell what's going on. But I think by the end of this year, we'll have it resolved. 2011.
Isn't that what we said last year, though?
It is, yeah. So if I keep saying that,
it'll keep not being resolved.
Maybe.
So
What's even...
Well, it'll be interesting future shows of TWiV.
That's right.
Yeah.
What's even more amazing is that there are people out there that don't believe HIV virus causes the condition.
Oh, thank you for bringing that up, Kathy. We got an email not too long ago from one of these AIDS denialists, right?
Yeah.
And he gave us a whole list of stuff that's never been proven, and therefore HIV cannot cause AIDS. So, yeah. I mean, we figured that it wasn't really worth going into. But in your view, what are the strongest points that HIV does cause AIDS? What's the best data?
Well, we can use drugs that we know specifically block HIV infection by very, detailed scientific well-studied mechanisms.
Mm-hmm.
And specifically those drugs that we know to disrupt HIV infection and HIV spread also stop the disease from progressing. So those people that take the drugs that are anti-HIV do not develop AIDS.
Mm-hmm.
And these are really specific medications that are designed with the HIV proteins in mind. Particularly the protease inhibitors, which were based on a crystal structure of the HIV protein, have really dramatic effects on viral levels in the blood, and also are really important to the cocktail that prevents the development of AIDS. Also, we can take simian immunodeficiency virus, which is very, very similar to human immunodeficiency virus, and put that into animal models and generate symptoms that are very similar to those that people get when they become HIV infected and develop AIDS.
Mm-hmm.
Also,
So
don't we have that experiment that has unfortunately been done in some human cases, like with lab accidents and needle sticks?
There have been some healthcare workers and one or two researchers who have picked up the virus from exposures, and the correlations, really good between becoming infected with the virus and developing AIDS in untreated people.
And the drug reverses dire illness.
Yes.
Right.
Yeah.
Which is, I would say, the hallmark of the whole thing, right?
So what sort of...
But my question back to the denialists would be, so what happened when they injected themselves with a pure stock of the virus?
Exactly.
Because they must have done that if they really believe what they're saying, or maybe they lack the strength of their convictions. I mean, if they really want to prove their point, that would be...
Well, I think they'd rather argue it on paper, Alan, right?
Well, but if they truly want us to believe this, do the experiment already.
I don't think that's ethical anymore. It's not the 50s.
If you're doing it on yourself...
I don't think you can do it on yourself. You need an
can't give
IRB approval.
You can't give yourself permission to disobey the law.
Rich, were you going to say something?
Yeah, I was interested in what sort of biosafety issues you confront, Kathy, in working with HIV.
So we usually work in a BSL 2+, it's called, or a BSL 3 laboratory setting, and HIV is not spread through the air, so we don't have to worry very much about aerosols. We wear gloves and we bleach all of our cultures before we dispose of them, and that inactivates the virus. And we just avoid using sharps or situations in which there could be splashing into somebody's eyes or mouth. That's the way that it's transmitted.
Right. So it's very much the same as what we do with pox viruses, except that, pox viruses, if you have an accident, you get over it.
Right.
With HIV, if you have an accident, you don't.
Right, but if people have accidents and then go get some treatment, then that reduces their risk.
Okay.
And it's a low risk from a needle stick exposure to begin with. I believe the numbers are about 0.3% transmission from a bloody needle stick.
And it's basically got to be a needle stick in order to get you infected. You have to actually get it into your bloodstream, right?
You have to get it into your bloodstream. So a needle stick or an abrasion, splashing onto an abrasion or splashing into eyes.
It would have to be like a fresh cut or splashing into your eyes. Okay.
Yeah.
It's not terribly infectious because even sexual transmission is like 1 in 200, I think.
Mm-hmm.
So do you think that there's an immune surveillance for infection in general that overcomes these low doses of exposure and prevents infection?
I'm sorry, can you repeat that?
Sure. I'm looking at a situation where it's dose dependent, okay? So below a certain dose, you don't get infected. Above a certain dose, you do get infected. Is it that kind of thing we're looking at, or is it inactivated virus and you only have a very few active particles at any one time, and so therefore...
I see.
See what I mean? So why don't these people with sticks in their fingers become infected? What's the reason for that?
Yeah, I don't know. It's I don't know if maybe there's we do know that there's a high rate of defective virus particles.
Right.
Maybe even a couple logs, with lab-produced virus, and so it may be that there's just not enough functional particles.
Do you ever get people who seroconvert but don't actually show up with active virus replication?
Yeah, there's a category of infected people called elite controllers.
Okay.
Right.
And they have HIV, they have antibody to HIV, but it's really, really hard to find the virus.
Right.
And they don't get sick from the disease.
Right. That was Al Saw's original group, by the way.
Yeah, they were elite controllers.
That's what he identified.
Okay.
These people that were exposed all the time. Sort of sex workers, etc., that never became infected.
Yeah, we talked about those on TWiV a long time ago. Some of them go along with certain HLA types.
The other thing I just wanted to mention is that I'm a personal friend of Irving Weissman, and I know that he is a good friend of David Baltimore, and I know that
they fish, right?
They fish a lot, but the other thing is that Irving Weissman is best known for the fact that he took mouse bone marrow cells and reduced the number of isolates down to 7 cells total, and with those 7 cells he could reconstitute the entire immune system of a mouse.
7. There are 7 genome types, Dickson.
No, we're not doing this.
This is amazing.
The 7th son of a 7th. But apparently only one of those cells could do it, but you couldn't get it below 7 and still have the cell there in the serial dilutions.
You're talking about reconstituting
the entire
from
immune
these
system.
precursor cells?
Correct. So he formed a company based on that from Stanford.
Yeah.
And I guess he made a lot of money.
He did make, and he has a nice fishing place in Montana or wherever.
Montana, yeah.
That's right.
Where he hangs out with David Baltimore.
He does.
Ted Turner also probably.
Yeah, that's right. And another good friend of mine is Roland Scully, who also worked on the B-cell maturation
This week
system.
in name dropping.
No. I was just saying that...
Well, Sean Morrison, who helped us with our mouse studies, did work with Irv Weissman. So.
Yeah. You learn a lot by going fishing with these people because we don't catch a lot of fish. But we do talk a lot while we do fish. So, nice way to learn virology.
Shall we do some email?
Sure.
Kathy, you want to stick around for email?
Sure.
Okay, so the first one is a response to a letter we got last time from a listener who asked whether endogenous retroviruses have provided some selection for the maintenance of TRIM5 alpha. So I asked Welkin Johnson about this, and he replied. He said, I get this question a lot, but never as well thought out as this. The listener clearly understands population dynamics better than the average virologist. However, I don't think endogenous retroviruses can possibly be a major selective force working on TRIM5 alpha. First, any endogenous sequence present in the genome must have gotten past TRIM5 alpha in the first place, so would have been pre-selected for resistance to the major alleles present in the host. Second, most ERVs probably go through a powerful selective filter for non-pathogenesis or non-functionality at the time of acquisition. If not, the first individuals to carry the ERV would suffer decreased fitness and be less likely to pass the ERV on as a genetic element to the next generation. Random mutations and silencing by methylation are more likely to deal with the ERV in a short evolutionary time period and more probable than a chance match between ERV target and TRIM5 alleles. The scenario posed by the listener in which different ERVs select for maintenance of different TRIM5 variants could work, but I suspect the impact of exogenous retroviral spread would outweigh by orders of magnitude the effects of rare activation of ERV, especially given these 2 points. To put it another way, a retroviral epidemic sweeping through a population of related individuals should have a much stronger effect than a rarely activated ERV appearing generations apart. However, the scenario might apply in the specific case where expression of the ERV had taken on a cellular function, with TRIM5 playing the role of a regulatory protein, but that would be an unlikely exception. All right, thank you, Welkin. So we were wrong. We said, sure, it could be.
I told you we were unthinking here.
Yeah. Well, you could either, if you say yes or no, you got 50-50 chance of being right.
That's right.
All right, the next one is from Jesse, who writes, thank you so much for your podcast. I am currently practicing as a small animal veterinarian while also studying towards a Master's of Public Health in the veterinary concentration at the University of Tennessee, Knoxville. My question is regarding the Wolbachia studies, which were discussed in episode 115. This is the release of Wolbachia-infected mosquitoes to try and stop dengue transmission.
Right.
Recent protocols for the treatment of canine heartworm disease suggest pretreatment with doxycycline to kill off the Wolbachia, which many of the Dirofilaria immitis larvae harbor.
Correct.
Did I get that right, Dickson?
Yes, you did. Yes, you did.
It is thought that Wolbachia may contribute significantly to the inflammatory response post-traditional treatment with melarsamine. So I wonder, how might the infection of an Aedes mosquito with Wolbachia affect the transmission of this nematode, which itself is infected with Wolbachia?
Maybe that's how the larvae pick it up.
Perhaps, as was suggested, this is an unnatural infection for these mosquitoes, so any released populations may die off. But what do you think?
Well, it's possible that... I don't know. No. I'll back off. I haven't thought about this long enough to give an answer. I must say that Onchocerca volvulus also has a Wolbachia. So Filaria appear to have this endogenous symbiont with them. And whether or not they're biologically important for the parasite to harbor, or whether they're just unfortunate passengers that cause pathology, which is targeted towards the Wolbachia, not against the parasite, which is quite interesting.
So this mosquito's got a nematode in it?
Yeah, that's how they pick it up.
No, but the nematode is mosquito-borne, is that right?
Yes, that's correct. So the larvae already have the Wolbachia in them because they're an endosymbiont within the parasite, so that as the parasite reproduces, they produce more bacteria as well, almost like mitochondria reproducing inside normal cells. So they wouldn't pick it up from the Wolbachia-infected mosquito. So
are all mosquitoes infected with this nematode?
Oh, no, quite the contrary. Very few percentages of mosquitoes that do transmit this are infected with this nematode.
All right, so I don't understand his question. How might the infection of an
Correct.
Aedes mosquito with Wolbachia affect transmission of this nematode, which itself is infected with Wolbachia?
Exactly. So I guess he's raising a question of, is it a good control measure to try to infect Aedes aegypti with Wolbachia to try to control the mosquito? That's his big question. So nobody knows what the answer is. But you know what? In the laboratory, you could actually do that experiment because these are easily controlled in the labs.
I guess I don't understand the nematode connection here.
Well, all I'm going to say is that if Dirofilaria immitis microfilariae already have Wolbachia in them as they're produced in utero in the adult worm, which lives in the heart of the dog, then a mosquito that's already infected with Wolbachia picking up a larva that's already infected with Wolbachia, those are two...
But how often is that going to happen in Malaysia where you're trying to control dengue?
Well,
it might happen?
In Malaysia, it could happen quite frequently because they've got a lot of filariasis there. And in this country, we have a lot of Dirofilaria. So we do have a lot of infection, but it's not human infection.
Well, you know what it comes down to is that biological control measures are always doomed.
You know, that's true.
Starting with the pox viruses in Australia.
No, there's no question about this because it reaches a balance. It's all about population dynamics. And I don't know any that's actually worked out good.
Well.
Or well, even.
Okay. Thank you, Jesse.
Wolbachia.
The next one is from Shama, who sent a link to an article called How the Pox Epidemic Changed Vaccination Rules in NPR. Hey, Rich, are you still there?
Yes, I'm still here.
I'm
looking at this article.
You're stunned?
No. What, you want me to talk?
No, you don't have to talk. It's okay. Never mind.
So this is a great, it's a really nice link. Actually, it highlights this book called Pox and American History, which I have not read. But this NPR broadcast is about this. And it really highlights the history of a problem we've been talking about, and that is how do you enforce vaccination?
Right.
Apparently back at the turn of the century, there's been resistance to vaccination ever since it was invented.
Right.
And for various reasons at different times. And apparently back at the turn of the century, when smallpox was really a problem and there were people resisting vaccination, there were troops of people that went around in various parts of the U.S. and forcibly vaccinated people.
That's right.
So this NPR report highlights some of that and some of the later evolution of sort of legislation to try and enforce vaccination. So
I understand.
Article.
I understand that there was a banker in Philadelphia during that major epidemic during the, I guess, Revolutionary War time, that tried to demonstrate why it was a good idea by he had 6 sons, and 3 of them received the vaccine and 3 didn't, and then he brought them all to a smallpox ward. And 2 of his 3 sons died that didn't get the vaccine, and none of the ones that got the vaccine died. That might be an apocryphal story, but I heard that. I thought from Elvin Cabot as one of his anecdotal stories that he used to give in the immunology course that he taught here. So, I mean, Jenner's original work in London, or in England, I should say, was accepted there, but the people would say, well, that's true for England. That might not be true for us.
Sure.
So now you have to re-demonstrate the same principle in another place. That's not uncommon for drug use and vaccines and all kinds of other things. So I understand why there would be a game changer on this one.
So it says there's this one the one's highlighted here. There was one episode in Middlesboro, Kentucky. This is at the turn of the century, where police and a group of vaccinators went into an African-American section of town, rounded up people outside this home, handcuffed men and women, and vaccinated them at gunpoint.
Wow.
Okay. It is a shocking scene that very much is at odds with our daily held notions of American liberty.
Right.
But it does highlight what is a continuing big problem.
Sure.
How do you ensure that people get vaccinated?
No, you don't
I
it
guess
at
that's
gunpoint.
Yeah, you don't do it at gunpoint.
Well, in New York City, they withhold school from the kids.
Well, that's the thing. But you can get around that, Dickson.
I know that. Homeschooling, it's called homeschooling.
No, you get an exemption. You can get a religious or a medical exemption for sure.
Right.
Yeah.
And then people argue, and it's hard to argue against it, that this is their right. But with public health, vaccines work when a lot of people get them, right? So it's a tough one. It's a very tough.
It's a misuse of religion versus the state's rights to.
Well, that's the only way they.
I understand that,
do
but
it.
I thought we separated those two things a long time ago.
I don't think so.
That's a separate podcast.
That's a separate one. Let's read one more email and then we'll move on. Next one is from Tom. Thanks for the great episode on HPV. I'm a big fan of the show. In general, it's been wonderfully helpful as a grad student working on a virology HIV project, and I recommend it to all my peers. And I have really enjoyed consuming your weekly picks as well, particularly the book recommendations. I feel that one important point was omitted during the discussion of the rationale of vaccinating men against HPV. In addition to serving as a vector for cervical cancer in women, men are susceptible to anal cancer linked to HPV. Rates of anal cancer are particularly high in at-risk populations like men who have sex with men. And it gives a JAMA reference. I don't know if the cost-benefit analysis has been performed for vaccinating to prevent anal cancer as it was with cervical cancer, but it is an important point that parents and individual patients should consider when thinking about getting vaccinated against HPV. Great episode, though. The HPV vaccine is such an important accomplishment in modern medicine, and I appreciate you highlighting it. Thanks also to Michelle Osborne for sharing her insights. Tom is an MD PhD student here at Mount Sinai in New York City.
Great. That
Cool.
is another thing we didn't really emphasize much on that HPV episode is that more and more HPV is becoming linked to various head and neck cancers.
Yes.
Okay?
Right.
Yes.
So it's showing up in more places than we originally suspected. Now, the types that are showing up in head and neck cancers, I'm not sure that they're the same types that are in cervical and anal cancer, but we now have the technology to make the vaccines, so maybe those will become important as well.
Yeah, my feeling about it is it's a virus. We know it's pathogenic. We have a vaccine against it. So what's the question?
Right.
But that's obviously not the prevailing view everywhere.
All right, let us move on to some picks of the week. Dickson, do you have a pick this week?
I don't.
All right, that's fine. You're not going to get your meal tonight, but
That's okay. I could use the weight loss. Maybe I should pick a book by
you don't have to. It's all right.
Mehmet Oz on You on a Diet, perhaps.
No. Don't pick Mehmet
Please don't
Oz.
do that.
Alan, what do you have?
I have it's a site called World of Viruses, and I actually came across it as I was starting to read Carl Zimmer's latest book, Planet of Viruses. And I thought it was a pretty cool site. What I thought was neat was they are making a genuine effort to explain viruses to the general public. They are supported, at least in part, by a grant from the NIH to do exactly this. And they've produced. So Carl's book, I gather, was part of that. They had him write the book. And then they've also produced a set of comic books about viruses, which I downloaded the iPod app, and you can read these comic books on your iPod or iPad, or I think you can read them online as well. And they also have a set of what they call radio programs, but they advertise them as CDs, or you can listen to them online. And just having a quick look at it, I thought it was a really cool concept of a way to get the word out. But this is very clearly aimed at the masses. It's aimed at reaching as many people as possible. And as I say, I've just started reading Carl's book, and it's not hardcore stuff. It's very accurate from what I can tell so far. But he's clearly aiming at a broad audience. Mm. So I think their approach in terms of that part of the presentation is about on target.
Oh, I see. I can read the whole comic online.
Yeah, or on your iPhone.
I'm just looking at the Curse of the Tree Man here that has a very seductive blue woman on the front.
Yes,
with a badge that says HPV on it.
Yes, HPV is very sexy.
Okay. I'm going to have to read this.
Yeah, the comics are well done. They're I haven't read them all, but I've flipped through a few, and they are well drawn. They got good artists to do these, and they I don't know. It seems like an appealing approach to me.
Rich, what do you have?
I have something a little lighter.
So you think?
Did we lose Kathy? Are you still there?
I'm still here. I'm
sorry. Go ahead, Rich.
So I found this on a friend's Facebook link. This is a little late for this season, but this is a YouTube video of some guy who goes by the handle Catamole, who likes to put music on YouTube, and I guess he writes these songs. This is a biologist's Mother's Day song. Okay, and it's a song all about maternal inheritance.
Cool.
And it's actually very well done. He obviously knows his stuff. And basically, he's celebrating his mother on Mother's Day because she gave him a little more than half of what he is genetically.
And all the mitochondria, of course.
Yeah, exactly. Okay, so he goes through all that. Nice. And if you go to his, he actually has his own sort of page or section in YouTube. If you go there, he describes himself as a science bard, among other things. Oops. Gotta turn that off. Describes himself as a science bard, among other things, as he's written a number of different science-based songs. There's one for St. Patrick's Day about beer that's really pretty good. Okay, and all about fermentation.
Or the lack of biodiversity in Ireland because there's no snakes.
I thought that was entertaining.
Nice, very good. Okay, my pick is a product called Magnet Balls, which Dickson has right in front of him here.
Oh, my God.
These are little magnetic balls that you can get in a variety of places, and Rich will tell you how addictive they are.
Yes, I didn't realize. I'm glad you put this up here. Ooh, I can get them in different colors, too.
I love the cat picture.
The cat. Oh, these are different things you can do with them?
Yeah, they're just there's a custom image from somebody who made a hat for his cat out of them. That's great.
You can make all sorts of things, but we have made some virus particles with them here.
We have.
And Dickson's playing with tobacco mosaic virus right now.
As we speak, although I don't smoke.
So they're $20 at Amazon and pretty cool.
They're pretty
cool. Yeah,
cool.
They are quite addictive. So I'm gonna get me some of these.
Yeah, different colors. You get silver, black, gold, platinum.
I think psychologists should get them and then allow their patients to play with them, and then whatever shape they come up with, they can start a conversation.
Well, when Rich was here, he played with them all the time.
I know he did. I saw him.
Yeah. Kathy, you want to pick anything?
Well, you told me I didn't have to prepare for this.
Okay. Are you reading anything good lately?
Well, I have a paper on my desktop by Jeremy Lubin, who you may know.
Oh, yeah,
we
sure.
do.
Yeah.
He's got a new paper out in Nature on TRIM5.
Yeah, that's a
Very
cool one.
interesting.
In fact, he's going to be here in New York I think next month, and we're going to try
nice.
and get him
I like Jerry.
into the TWiV studio to talk about that. He used to be here, at Columbia.
I know. I met him there once when I was interviewing for a job at Columbia, actually.
He's coming back to the U.S. He's in Geneva at the moment. He's coming to UMass Worcester, I think.
Really?
Oh,
Really?
I know where that is.
Hey, it's not far from you.
Yeah,
That's
actually,
central
my part of the
Massachusetts.
state is often referred to by people in Boston as somewhere out west of Worcester.
That's
That's
right.
West of Worcester.
All right, thanks. Jeremy Lubin's TRIM5 paper. That's pretty cool. We also have a listener pick of the week from Chris. Periodic Tales, a cultural history of the elements from arsenic to zinc.
I read the review of that.
Yeah, it tells great stories of the history of discovery and uses of all the elements. Great read.
Yeah.
Excellent. That
Nice.
sounds good.
That's
Yep.
a good one.
Good stuff.
Thank you, Chris. And that will do it for another TWiV. Now, I just want to tell everyone next week, as the floodwaters approach New Orleans,
Yeah.
the ASM will be having its general meeting, and we'll be doing live TWiV and TWIM from the meeting. And you can go to virology.ws for details about that. You can find TWiV at iTunes, at the Zune Marketplace, or at twiv.tv. And there's also an app for your iPhone or Android device that you can find at microbeworld.org/app. And do send us your questions and comments to twiv@twiv.tv. Kathy, thank you so much for joining us.
It's my pleasure. Thank you
it
for
was
inviting
great.
me.
It was really great.
Yeah,
Thank
these
you.
are-
Yeah,
Absolutely.
great to have you.
University of Michigan, and perhaps Rich and I will see you there sometime in the next
I hope
year.
so.
That'd
I
be
hope
great.
so. That'd be good.
Dickson, nice to see you again.
It's nice to be here. Always.
Verticalfarm.com. Rich, thank you.
You're quite welcome. Always a good time.
University of Florida at Gainesville. And Alan Dove, thank you.
Always a pleasure.
Are you west of Worcester?
Yes, I'm west of Worcester.
And you're also at alandove.com.
That is correct.
And I'm Vincent Racaniello at virology.ws. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.